The abstract I presented at the EHA 2020 meeting was a study that we looked at in older patients with acute myeloid leukemia who were ineligible for intensive chemotherapy, implementing a novel combination of azacitidine with a CD47 antibody called magrolimab. This study was based on extensive preclinical data that has been done and published by the group at Stanford, looking at the use of CD47 blockade in patients with acute myeloid leukemia. CD47 is a very important immune checkpoint present on the surface of macrophages that, when engaged, inhibits the macrophage from performing their routine function of phagocytosis of foreign cells, which includes tumor cells. And the preclinical data showed that by blocking CD47 using magrolimab, which is a IgG4 CD47 antibody, we could get a high degree of activity, especially a high degree of synergy in combination with azacitidine in preclinical acute myeloid leukemia mouse models. This led to the combination of the azacitidine with CD47 antibody in the ongoing phase 1b study. The study has two arms. One of them is looking at patients with acute myeloid leukemia. These are patients who are older and considered to be not suitable for induction chemotherapy, either because of age alone or because of other comorbidities that would put them at high risk from toxicity, mortality from the induction chemotherapy. The other arm is looking at patients who have frontline myelodysplastic syndrome. This included patients with intermediate high, very high IPSS risk with MDS. And I'll be focusing on the AML data. My colleague, Dr. Salman, presented the frontline MDS data, and both these data sets are looking very encouraging at this time point. So in acute myeloid leukemia, one of the reasons we think that there is a distinct synergy between magrolimab CD47 antibody and azacitidine is because azacitidine actually has been shown to upregulate prophagocytic signals on the surface of leukemia cells. This includes signals such as calreticulin and other prophagocytic signals, and we're looking at this in detail on the ongoing study. And usually in the presence of the upregulated prophagocytic signal with azacitidine, there was a much higher synergy and activity with the CD47 antibody. So at this time, we had a total of 29 patients that were evaluable at the cutoff for this abstract. And this was enriched by older patient population. Median age was 75, and the upper age extreme was 89. So these were quite old patients. Also important among the patient characteristics was that we had a especially high predominance of patients with poor risk cytogenetics. 72% or 3/4 of the patient had poor risk cytogenetic per the ELN, and about half of the patients had TP53 mutation. And this was partly by design. About 5-6 months ago, when we started seeing encouraging responses and duration of responses in TP53, we amended the study to actually only focus and enroll frontline TP53 mutated AML, which is why we see kind of much more TP53 AML in this study. The safety is probably the thing to me that is most exciting at this time about this agent, given that we have kind of early follow-up. The combination in general was very safe. In fact, I would say very similar to our experience over the last decade with azacitidine alone. We did not see any immune-mediated toxicity such as organ inflammation, immune itises. We did not see any severe or prolonged or unexpected cytopenias. Now, the one thing that we do see, which was kind of predicted and expected based on the preclinical data, was an on-target anemia. And this is because CD47 actually is heavily expressed on the surface of older red cells. And so when you give the CD47 antibody, there is an initial clearance of the older red cells. And then the younger red cells, the reticulocytes that come in, often express low or no CD47. So to try to mitigate this anemia, we've kind of used a intra-patient dose ramp-up escalation schedule. And with that, we did see anemia, but it was transient, reversible. No patients had to come off study. There were no severe effects from the anemia, and it was quite manageable. And usually by day 12 to 14, the anemia regresses consistently in most patients. Also in the safety, especially in this high-risk group, that I think is really most critical, the 60-day mortality was 0. And this was true in both the AML-MDS studies. So aside from response and survival, the third parameter we usually consider important in older AML is 60-day mortality, and I think that was very favorable in the study. From efficacy point of view, we had 25 patients who had completed the time period of at least 2 cycles. A couple of patients are still too early at the time of the data cutoff. And the overall response rate was 64%, which included a true CR/CRI rate of 56%, and then a few additional MLFS, PRs. This is encouraging, definitely better than the expected CR rates of 15 to 18% that have been shown with azacitidine decitabine alone in phase 2, phase 3 studies in similar AML population. I think more importantly is the durability of the response. At this time, we see that the median duration of response has not been met with a median follow-up of about 8.5 months. And also importantly, we looked at some deeper response parameters, including MRD by flow cytometry and cytogenetic responses. And we see that about 50% of patients have a cytogenetic response and 50 to 60% have MRD negativity, indicating that these are in majority quite a deep response. We looked at survival. The 6-month projected survival is 91%. And at this time, the median survival has not been reached. And the survival curve looks quite encouraging. The last thing, which I think is probably going to be very important for the development of this drug in AML, is that we actually did have a good number of TP53-AML patients in this study. There were 12 in total out of the 25 evaluable. And among the TP53, the CR/CR rate was 75%. Median duration of response not reached at this time, with a median follow-up of about 8 to 9 months. And this compares favorably to azacitidine venetoclax, which is the new standard for frontline older similar AML, where the response rate, CR/CRi, is about 47 to 50%, and the median duration of response is around 4 to 5 months. So again, early days, we need to build on this. The study is only enrolling TP53-AML. We should have more patients enrolled by the end of the year, and hopefully we'll get an update. But the future development of the azacitidine magrolimab in the AML is going to be focusing heavily on the TP53 mutated AML population. And hopefully we will have some interesting activity in this subset, which continues to remain probably the most difficult subset in AML and MDS. Thank you.